Yes.
Ma'am, we are live.
Okay, sir. By taking the permission of Honorable Principal and Honorable Chief Guest of today's webinar, I am starting webinar. Good afternoon and warm welcome to all in today's state level webinar on contribution towards click chemistry, which is organized by the Department of Chemistry and Internal Quality Assurance Cell, Bharati Vidyapith's Dr. Patangrao Kadam Mahavidyalay, Sangli, on the occasion of National Chemistry Week. National Chemistry Week is an annual event held in the United States to raise public awareness of the importance of chemistry in everyday life. It is coordinated by the American Chemical Society. National Chemistry Week is celebrated every year in our college by various activities. For this year, it is being celebrated from 16 October to 22 October. Today's webinar is the part of this event. The president of today's webinar, Honorable Principal Dr. D.Z. Kanse sir, former member, Management Council, Shivaji University, Kolhapur, and chief guest, Honorable Professor Dr. B.B. Shingate sir, are present online in front of us. Now I welcome to President Honorable Dr. D.Z. Kanse sir, chief guest, Honorable Dr. B.B. Shingate sir, all faculty members and students. Now I invite to Dr. A.R. Suple sir for offering the introduction. Dr. A.R. Suple sir.
Thank you, madam. Good afternoon to all respected dignitaries present over this platform and all the participants. I welcome you all for state level webinar on contribution towards click chemistry organized by Department of Chemistry and Internal Quality Assurance Cell of our college, Bharati Vidyapith's Dr. Patangrao Kadam Mahavidyalay, Sangli. As all of we know, in last week we have heard the news regarding Nobel Prize announcement for various subjects, and we know that the Nobel Prize 2022 in chemistry has been awarded to three scientists, and this one is for their invention for the development of click chemistry and bioorthogonal chemistry. So, as a chemistry student and as a science student, we must understand what is this concept and this invention, what are the scenarios of this invention in the future. So we have invited today's chief guest, Dr. Shingate sir, who is associate professor in Aurangabad University, to deliver his talk on this particular subject. I will introduce him in brief. Dr. B.B. Shingate is working as associate professor at Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. His specialization is organic chemistry. He has obtained his PhD degree from NCL Pune. He has total 20 years of research experience and 14 years of teaching experience. He has recipient of many awards, that is Best Chemistry Professor of Year 2020 from Global Teaching Excellence Award, recipient of ISCB Best Teacher Award in 2018, Indian Society of Chemists and Biologists. Then he also served as a Bentham Ambassador. He also a recipient of Research Professor Award of Aurangabad University. He also recipient of Ideal Teacher Awards. He also recipient of Indo-US Research Fellowship in 2013 and Shikshan Pratibha Puraskar from Aurangabad University. He is working in the area of synthesis of bioactive compounds and new synthesis methodologies. He has delivered 85 talks as a research person in various conferences and seminars, symposia. He also delivered a number of talks at various university and colleges. He also served as member BOS at universities and autonomous colleges. And 7 students have been awarded for their PhD degree under his able guidance. He also worked as guest. He is also working as a guest editor, associate editor, and editorial board member for various journals. And he has also to his credit, having citations 3447 and having H index certificate. He is expert in this particular field, and we expect a very fruitful session at this afternoon. So, on behalf of college, I welcome you, sir, for this particular webinar. Uh, for this session, we have our principal of Principal Dr. D.G. Kanse sir as a chairperson. He is
Now I requested Dr. B.B. Shingate sir to deliver his talk.
Okay.
B.B. Shingate.
Okay. Yes, Lohar sir, can we start?
Yes, sir.
Yes. Okay.
Yes, sir.
Thank you very much for your nice introduction. And first of all, I would like to congratulate. Am I audible?
Yes, sir.
Yeah.
Yes, sir.
Firstly, I would like to congratulate the college, Bharati Vidyapits, Dr. Patangrao Kadam College, Sangli, for organizing such an event that is National Chemistry Week. And today is the first day, and it's a great opportunity to me to share my views regarding recently the Nobel Prize was awarded to the three peoples from the chemistry discipline. And on the occasion of that Nobel Prize in chemistry, just I am presenting over here our contribution in terms of the concept that is a click chemistry. First of all, I would like to thank to the principal of Bharati Vidyapits, Dr. Patangrao Kadam College, Sangli, as well as faculty members from chemistry department, as well as other departments, as well as my dear participants. I am very much thankful to all of you and giving me an opportunity to share my views regarding click chemistry, our contribution. Just I will present my presentation in front of you. Sir, I am presenting.
Yes, sir.
Just. Sir, share screen na? Hello?
One day,
One day,
sir.
sir.
Yes, sir.
Right.
Here is screen. Yes, sir.
Share screen or
Yes,
slides.
Uh, present. First, we will click on the present,
Yes.
and after we will click on the share screen.
Yes.
And
Then
afterwards, we will click on the window instead of entire screen.
okay. Screen sharing. There is no window.
On the right hand side, sir, there is a window option at the middle. Entire screen
No.
window and the Chrome tab.
No,
Window
sir.
is
See, screen sharing tips like that way. Screen sharing is easiest with 2 months. Screen sharing works
Sir,
best
skip
of.
that. Sir, skip that one.
Yeah. Then
Entire screen and second option is window.
screen share. See, after project screen share screen. Then share screen again it is coming.
Yes.
Uh,
And there is 3 options are there. Entire screen as we did yesterday.
oh yeah, yes, sir. Entire screen window. Yeah.
Yes, you sir, just simply click on the window and then we will proceed.
Yeah. Now, is it visible to you?
Sir, I will put it on the screen. Wait.
Yeah.
Yes, sir.
Whether it-
It on the floor. Please continue.
Yeah. Can you read the title?
Contribution towards the click chemistry.
Yes. Shall I start?
Yes,
Yes, sir.
Yeah. Thank you for your nice technical support, sir. And
Welcome, sir.
today I will talk on contribution towards the click chemistry, which is a hot topic from last 20 years. And I will present our contributions in terms of click chemistry. Before going to talk on click chemistry, why we have chosen this particular area as a research. That's why, first of all, I will take the opportunity to discuss about the different causes of death in India. As we know, as we are witnessing about the peoples are suffering from different kinds of diseases, and the net output of those diseases, some of the diseases came in the death, particularly in India. The first and most important disease is related to heart disease, and nowadays it becomes common, and uh, that is a stroke, heart attack we are calling for that one. And the second disease is related to chronic obstructive pulmonary disease. Third one is a diarrhea. Fourth one is a stroke, brain stroke. Then fifth one is a lower respiratory infections. As we have witnessed that in COVID-19, most of the people, they are facing for the problem of respiration. Then the sixth and most important disease is related to tuberculosis, which is an infectious disease. Then neonatal preterm birth, as we people have observed that many more births are preterm births. Then self-harm by means of different causes are there for self-harm. Then road injuries, a lot of road injuries happens in India, and other neonatal conditions as well. In addition to this one, these diseases, we have witnessed that some other diseases may cause death in India. That is a swine flu in last 10 years, in the last decade as well. Then dengue. Then from last 3 years, we have witnessed that this coronavirus, COVID-19 pandemic, and due to this COVID-19 pandemic, billions of people's cause into deaths, as well as HIV and AIDS, such causes of death in India, we have witnessed that. And the major cause of death in India from last 30 to 40 years till today, one of the most important disease, that is a tuberculosis. That is a tuberculosis, and just I will give you highlights of this tuberculosis. Because of not only this click chemistry is useful for synthetic organic chemist, but also medicinal chemist, then material science peoples, polymer science, then biological sciences, as well as pharmaceutical sciences. And our interest to design and develop new chemical entities for curing one of the disease which India is facing a lot, that is the tuberculosis. And we know from last 30 to 40 years, at least one patient from small villages of Maharashtra and India, at least one patient in small villages we have witnessed, observed. And because of that TB, later on some years, after taking some medicines also, they are suffering from that TB, and later on they may come into death. Tuberculosis is one of the top 10 causes of death worldwide, and as per the data by WHO, that is World Health Organization, for the year 2015, around 10.4 million people fell ill with TB and 1.8 million died from the disease. Means around 20% patients from tuberculosis, those who are diagnosed, around 10.4 million, 1.8 million, means 20% people are died because of this tuberculosis every year. Over 95% of TB deaths occur in low and middle income countries. See, that is alarm to all of us. 6 countries account for 60% of total, with India leading the count. Means majority of the patients belongs to India. See, with India leading the count, followed by Indonesia, China, Nigeria, Pakistan, and South Africa. And we, all these 6 countries belongs to low and middle income countries. And from last 70 to 80 years, lot of developments in various kinds of drugs so far. And just we will see the journey of those drugs prescribed by the doctors. for the patients of tuberculosis. See, the first drug was developed in 1943, around 80 years before. That is a para-aminosalicylic acid. Until till date, this particular drug is prescribed by means of doctors. Then later on, this is streptomycin, which is an antibiotic, which has made a remarkable progress in the curing of tuberculosis. After 3 years, this is thiacetazone by Domak in 1946, and from 1952 to till date, means even though after 70 years, this particular isoniazid drug is given to the patients of tuberculosis. Then pyrazinamide in 1952, in the same year, because that was the era of design and development of new anti-tubercular agents, that in the 20th century of sixth decade, that was the era of design and development of anti-tubercular agents, because that time lot of patients was there. In 1952, pyrazinamide, very small drug, isoniazid is also very small drug. And in the year of 1952, 1957, rifampicin was developed by means of this Margalith, and this rifampicin has totally changed the strategy in the chemotherapy of tuberculosis. In
Sir,
1960, yes.
will you please click on full screen?
It's full screen is there, sir. For me, full screen is there.
Full screen is here. But what
Yeah,
happened actually, sir, on our side it will stuck actually. So
I
what
don't
we will
know.
do? What we will do, sir? I will remove this screen and I will add it again.
Okay. Shall I stop for some time?
Wait. Sir, you will exit presentation from your side presentation and you will add again.
Again.
I think
Okay.
some is network delay from your side for the presentation.
But sir, it's not visible. I think.
Yes, sir. Yes.
Whether it is from my side or from your side.
Sir, I think this is from your side because presentation is gets stuck.
Okay. But network, I have network, full network, no issue.
No, sir, not the network. You will just remove from presentation. You will exit the presentation.
What happened actually? Slits are yes.
Yes, sir.
I
Right
see.
now going well.
Just
Oh.
slightly.
Yeah, it is not a presentation mode. It is a simple mode, sir. Now.
Yes, sir.
Okay,
Yes.
I can go through this way also. No problem. I don't have any issue. I will continue. Okay.
Sir, will you please click on the full screen from the slideshow?
Yeah. Now, yeah, it is a full screen now.
It's full screen.
Yeah.
Yes, sir.
Now it is visible to everyone.
Yes, sir.
Yeah. We will continue. Yes.
Students are visible.
Sir,
Auditorium?
right now students are watching all these things.
Yeah. Shall we continue, sir?
Yes, sir.
Yes.
Yeah. As I'm talking about this tuberculosis, and I have told you that India is the leading count in terms of this patients belongs to tuberculosis, and we are talking about the journey of various kinds of anti-tubercular agents like para-aminosalicylic acid, streptomycin, thiacetazone, isoniazid, pyrazinamide, rifampicin, as well as ethambutol came in the market in the year of 1961, and followed by this fluoroquinolones, and later on this again conjure of rifampicin was developed. Then just I will show you the different kinds of drugs prescribed by the doctors to the patients of tuberculosis, and there are three categories of the drugs. Which one is a first line drug, second line drug, and newer second line drugs? In first line drugs, isoniazid, rifampicin, ethambutol, pyrazinamide, and streptomycin. And in second line drugs like thiacetazone, para-aminosalicylic acid, kanamycin. In the third class of the drugs, that is newer second line drugs, that is ciprofloxacin, ofloxacin, levofloxacin, as the clarithromycin and azithromycin. These are the higher antibiotics we know very well. And nowadays, doctors are prescribing, if even though patients is suffering from first stage of tuberculosis, then isoniazid, rifampicin, ethambutol, and pyrazinamide. Combination of these four drugs doctors are prescribing. But if the patient is suffering from multi-drug resistant tuberculosis, see, that is the second step of the tuberculosis, and very few cases, means only 5 to 10% of the total tuberculosis patients we are suffering from total drug resistance tuberculosis. Multi-drug resistance tuberculosis, and that time combination of first line, second line, as well as newer second line drugs were prescribed. For total drug resistance tuberculosis, then combination of all the first line, second line, and newer second line drugs doctors are prescribing for the patients, those who are suffering from total drug resistance, that is TDR, as well as XDR is also there. Means, see, it is a simple infection. Infection occurs from different sources, not only from the ear but also from the patients, those who are suffering from tuberculosis. It may comes the viruses of that TB. Now I will show you, being the organic chemist, medicinal chemist, we have to look for the structures of those anti-tubercular drugs, and just I am showing here representative drugs and their structures. Simply see, this isoniazid is nothing but isonicotinic acid hydrozide, which is the smallest drug used for the tuberculosis patient. This pyrazinamide, ofloxacin, kanamycin, rifampicin, ethambutol, and see, this particular drug has made a drastic change in the treatment of tuberculosis, that is bedaquiline. And as per the experts in the tuberculosis, they are saying that next 10 years, this drug can survive the patients of tuberculosis. And it is a simply, yes, yeah.
Sir, slides are
Yes.
not moving.
Yeah,
Slides are not
visible.
changing.
Principal sir. Hello.
Hello.
Yes, sir. That's why I'm raising this point. Sir, what we will do, we will just shuffle the slides for once.
Now
So
slide
this
3
slide.
is going on. Synthesis slide 3 is going on. Is it? Is it
Uh,
true?
I
No,
think they
no,
are
no, no.
modified.
This is
No.
second slide, this is third slide, this is fourth one,
No,
fifth
no,
one,
sir.
and this is the sixth one, sir.
No, sir.
No. What happened actually on the screen? The third
Yeah.
slide is.
Is going on. Shingate sir, what we will do right now, I am removing your presentation from the our floor, and we will
Ah.
add it again
Okay,
by clicking on the present.
okay. Shall I do once, and can I join once again?
No. There is a not from the your issue. This is the issue of the presentation. Wait.
Okay.
Sir, click on the present.
Yes.
Click on the share screen.
Already it is there, no, sir?
You will click on the share screen.
Okay. Again, present. Share.
Sir, instead of window, you will click on the entire screen. Select that screen.
Share screen. Okay.
Select that screen, and then you will click on the share.
Now, is it visible to you?
Wait. I will. Yes, sir. Right now it will going very well.
Yeah. That is the problem of sharing only windows and sharing full screen.
Sir, what we will do? You will shuffle the screen. We will check it, and then we will proceed.
Okay.
Just go up and down on the
Yeah.
screens.
Yeah.
Yes. You're
Shall
doing well.
I make it in presentation mode?
Sir, what you will do? You will go to the slideshow and you will make it a full screen.
Yeah.
So it will. Yes, sir. This is good.
Yeah.
You will continue.
Yeah.
Okay, sir.
Now
Sir, sorry
there
for
are
the disturbance.
okay. There are the structures of the drugs which are used for the treatment of tuberculosis, that is isonicotinic acid hydrozide, INH, pyrazinamide, ofloxacin, kanamycin, rifampicin, ethambutol, and bedaquiline. What I am talking about, this bedaquiline, this particular drug can survive for next 10 years as per the experts in the design and development of new anti-tubercular agents. Not only this one, but also another important disease which India is facing a lot, that is a fungal infections. And in the days of this rainy season as well as in winter also, most of the people, they are suffering from fungal infection. Different types of fungal infections are there. As we know, there are two types of fungal infections, and to cure those fungal infections, different kinds of drugs are used. Different types of antibiotics are used. For example, amphotericin B, nystatin, griseofulvin. Then second category of the drugs, that is azole antifungus and ketoconazole, miconazole, itraconazole. Such kind of drugs doctors are prescribing to us. Not only azole antifungus, but also there are some category of allyl amines, that is terbinafine, generally used to cure fungal infections, and pyrimidine based. This pyrimidine based drugs are also used to cure fungal infections, and the representative example is flucytosine, very small drug used to cure fungal infections. Now, our main focus is related to azole antifungus, and these azole antifungus are not natural products; they are the synthetic compounds. And there are two types of azole antifungus. One is imidazole based, and another one is triazole based. That triazole and imidazole based antifungal drugs inhibits the enzyme cytochrome P450 14 alpha demethylase, and this particular enzyme converts llanosterol to ergosterol and is required in fungal cell membrane synthesis. These drugs also block steroid synthesis in human. Okay, now see, as I have told you about the two categories of antifungal agents based on azoles. The first one is the imidazole based drugs like miconazole, ketoconazole, then bifloconazole, mebendazole, etc. These are the imidazole based drugs. And the triazoles, these are newer, less toxic and more effective. And see, the examples are fluconazole, rauconazole, itraconazole, voriconazole, posaconazole. And most of the researchers from synthetic organic chemistry, they are focusing on the design and development of new heterocycles which may contain imidazole skeleton or triazole skeleton. Means at least 2 nitrogen containing heterocyclic compounds or 3 nitrogen containing heterocycle compounds. And as I told you that regarding the triazoles, as they are the newer and less toxic and more effective, and the structures of these triazole based antifungal drugs, fluconazole, voriconazole and itraconazole. In their structure, see, there is a triazole core, and see the arrangement of nitrogen in the triazole core is at 1 to 4 position. In all the triazoles, here also 1 to 4 position, here also 1 to 4 position, here also 1 to 4 positions. It means that whatever triazoles are available in the market as a drug, that is a fluconazole, miconazole, itraconazole, voriconazole, these triazole based drugs are having 1 to 4 triazoles. Means the arrangement of nitrogens is at 1 to 4 positions. Now see, as we were talking about from last 1 week, from last 10 days, the Nobel Prize was declared on the date 5th October 2022. And these three peoples, Professor Caroline Bertozzi from Stanford University, USA, Professor Morten Mendel from University of Copenhagen, Denmark, as well as Professor K. Barry Sharpless from Scripps Research Institute, California State, United States of America. These three peoples have received a Nobel Prize in chemistry for the year 2022 for their contribution in the development of click chemistry, and bioorthogonal chemistry. And we all have witnessed that in the last week, everyone is talking about the Nobel Prize in different disciplines, that is the physiology or medicine, physics, chemistry, then economics, like that way. Different peoples have received Nobel Prizes. But in terms of chemistry, being the learner of chemistry, we should know about their contributions, in which field they have contributed a lot, and why they have received, why they have nominated for Nobel Prize, and how they have received a Nobel Prize for their contributions. And these three professors have received a Nobel Prize for their contribution in the development of click chemistry as well as bioorthogonal chemistry. See, as I have told you that these three people, Bartolozzi, Meldon, and Sharpless, in the year of 2000, Professor Sharpless have coined the concept of click chemistry, and meantime, in the same year, Meldon came up with special chemical structure called as triazole. And later on, in the year of 2003-4, Professor Bartolozzi took the next step of design and development of this click reaction that could work inside the living system. Means Professor Meldon and Sharpless, they have done the reactions in two different chemicals. However, Professor Bartolozzi have used one substrate from the living organism and another substrate from chemical part. And later on, C has labeled as that is a bioorthogonal reactions. And that's why click reaction came into the picture in the year of 2001 and called as click chemistry. And Professor Bartolozzi, Caroline Bartolozzi, have coined the term bioorthogonal chemistry. And see, this click chemistry is nothing but reaction between terminal alkyne and terminal azide. In between some catalyst, copper based catalyst, is copper catalyzed azide alkyne cycloaddition reaction. It is a simply cycloaddition reaction between azide and alkyne. Sometimes alkynes, not only from terminal alkyne, internal alkynes are also used. In presence of copper catalyst, different types of coppers were used, and very first time Professor Sharpless and Meldon have used copper sulfate as a source for the activation of alkyne, for the activation of terminal alkyne. And later on, different forms of copper people have used for the design and development of 1,4-disubstituted 1,2,3 triazoles. Okay, later on, copper acetate, copper chloride, copper oxide, copper nanomaterials, etc. Different forms of copper. Not only copper, but also some other different metals have also used. But the problem of regioselectivity was there. Regioselectivity. And only copper can use regioselectively 1,4-disubstituted 1,2,3 triazoles. See, as I mentioned that regarding this bioorthogonal reactions, it is carried out in the living organisms or in the living cells. That's why Professor Bartolozzi's contribution is a lot, means a direct applications of that click chemistry for living organisms, and very few reactions are there which are possible to carry out on living cells. Okay, this particular slide we have witnessed in the last week for several times, as well as I mentioned that it is nothing but the reaction between one substrate which contains terminal azide, another substrate which contains terminal alkyne. When comes in the contact in the presence of copper catalyst, and formation of 5-membered highly stable heterocycle, that is a 1,2,3 triazole. And see the arrangement of this 1,2,3 triazole. See, this part is from alkyne and this part is from azide. Similarly, Professor Bartolozzi have taken the advantage of this particular reaction and highly strained cyclic alkyne, that is cyclooctane, is used to carry out this bioorthogonal click reaction, bioorthogonal chemistry reaction by using azide. This azide, see the difference between click chemistry and bioorthogonal chemistry, even though the substrates are somewhat similar, that is azide and alkyne, but this bioorthogonal chemistry is without catalyst, without using any kind of metals over here. Means Professor Bartolozzi have avoided the toxic materials. Here the copper is a somewhat toxic material. That's why see, and simply when these two substrates are coming together, see over here, one reagent, another reagent, that is azide and alkene, they are coming together and formation of carbon nitrogen bond, and is nothing but click. It is a simple term. It is a simple term, click. Okay. Now, again, it can be explained like this way. This terminal azide, terminal alkyne, in presence of copper catalyst, formation of regioselectively 1,4, see 1, 2, 3, 4, 1,4 disubstituted, 1, 2, 3 triazole formation by means of copper catalyst. Okay. See, as Professor Sharpless have coined this click chemistry, and he has received a second Nobel Prize for his development of this particular click chemistry reaction. Prior to that, in the year of 2001, Professor Sharpless have received a first Nobel Prize for his contribution in asymmetric oxidation reaction. That is a Sharpless asymmetric epoxidation, asymmetric dihydroxylation, asymmetric amino hydroxylation reaction. And Professor Sharpless is the fifth person to win the 2 Nobel Prizes. He is the fifth person to win the 2 Nobel Prizes for his great contribution in terms of synthetic organic chemistry, and his chemistry is a highly useful chemistry. Okay, as I mentioned that it is the reaction between terminal azide and terminal alkyne in presence of copper catalyst. It gives a regioselectively 1,4 disubstituted 1, 2, 3 triazole. As mentioned by Professor Sharpless in his 2001 paper, very first paper came in the Anglo-Chem International Edition, and fortunately his author has delivered expert talk on the occasion of Nobel Prize on 5th October. Professor Finn was there, and he has presented this particular click chemistry on the occasion of Nobel Prize ceremony. And in that particular paper, what they are saying that it is a simple name for a set of reactions. Click chemistry is a simple name for simple set of reaction. The core of the click chemistry concept is the cultivation of a small set of reactions that are extremely ideal for working with organic molecules. Okay, means 2 organic compounds are coming together by means of copper catalyst. See, Sharpless described this in his famous 2001 paper. Then a click reaction would be modular. It is modular, wide in scope, means substrate scope is there. There is no any side reactions takes place, give very high yields, generate only inoffensive byproducts easily removed. As well as I want to tell you, it is a 100% atom economy reaction because it is a simply cycloaddition reaction like a pericyclic reaction. It is a highly stereospecific, it is a highly regioselective also because it gives 1,4 disubstituted 1, 2, 3 triazoles are there. Required simple reaction conditions. We will see later on how simple reaction conditions can be used to design and development of such kind of 1,4 disubstituted 1, 2, 3 triazoles. Requires readily available reagents and starting materials. See, these kinds of reactions can be carried out at any place, even though at room temperature, just only by simple stirring. Required no or benign solvent or easily removable solvent. See, easily removable solvents are used sometimes without solvents because many people have carried out these click reactions in ultrasound or microwave oven as well without using any kind of solvents. And most important thing is requires simple product isolation and purification. No flash column chromatography, simple column chromatography, or simply you can do purification of the product from the unreacted starting materials by use of crystallization also. Juice products that are stable in physiological conditions means these triazoles are highly stable under acidic as well as basic conditions. As I mentioned that it is a 100% atom economy as well as have a high thermodynamic driving force or highly stable even though at high temperatures also. And they are the similar to amides as well. That's why they are called as bioesters of amides. And we have witnessed that many more drugs they contain amide bonds in their structures, and it works like amide. These are triazole system. Okay, see how this simple reaction, click chemistry reaction can start from simple alkenes See here, just one of the important review article they have presented in this way. And even though in that Nobel Prize ceremony, one of the participants have asked the question regarding whether this reaction is a scalable reaction or not. Whether this reaction is a scalable or not. And here is the best answer for that particular question. Even though some of the reactions can be carried out in the lab only, and those reactions are not as scalable reactions. That's why, see, the way how they have contributed in terms of large scales also, even though at 20 grams, 30 grams, 40 grams, and some of the industries, they have started the business of development of triazoles also. Okay, see over here, simply here alkenes were taken, then epoxide, then opening of the epoxides by means of azide, and see, those azides were subsequently converted into the 1,2,3 triazoles, and see the scale of the reactions, 22.4 gram scale. Then from this to this, some of the transformations were there. Again, the scale was 16.7, and furthermore, there are some transformations. Again, 18.7 gram scale. Here also, same way they have carried out the similar type of large scale reaction, like that way. Okay. Then, why Nobel Prize was given for the year 2022? See, as I have mentioned that in the year of 2001, the first paper came by Professor Sharpless, and later on, see, in the year of 2010, the number of papers are 753, and the data was taken through the Scopus on October 11, 2022 at 5 p.m. And the total number of publications that time 17,665 are related to the use of click chemistry in terms of synthetic organic, material science, polymer science, then pharmaceutical science, drug, as well as the biological sciences. As I mentioned that in the year of 2010, the number of papers were 753. For the year 2016, 1,400. For the year 2021, 1,300. And for the year 2022, around 937. There are 3 more months are to go. And definitely the number will increase now. And many more researchers will start working on the click chemistry from this particular moment. And fortunately, we people are working from last 10 years. We are not the inventors, but we are the followers. From the year 2009, we have begun our motto of design and development of new chemical entities through click chemistry approach. Means just we are the followers, not the inventors. See, after seeing the number of publications, somebody can ask the small question related to whether some of the drugs are available in the market to cure some of the different kinds of diseases. Yes, there are few examples based on 1, 2, 3 triazoles, and just I'm showing over here. These are the potential pharmaceuticals. Just abbreviation I can take CAI, which contains 1, 2, 3 triazole in their structure, and such type of molecule was synthesized by means of click chemistry using this terminal azide and this this kind of alkyne can be used. Here also, see, particularly this azide is used for the development of this TSEAO molecule, as well as this cephalosporin based cephalosporin moiety, which is an antibiotic and having a beta-lactam core and having this 1, 2, 3 triazole skeleton over here, as well as this tazobactam ring. A tazobactam compound possesses 1, 2, 3 triazole in their structure. See, till today, there are n number of molecules were synthesized by means of synthetic organic chemists, medicinal chemists, polymer chemists, material science peoples have synthesized and studied their different kinds of properties. As per the Sharpless, it should not be the development of library of molecules, but you have to learn the properties of those compounds, organic compounds, and organic chemists can make a better structures of different kinds of organic molecules, and later on it can be studied that different kinds of, it may be some therapeutic properties or some other kinds of properties of those synthesized molecules. Okay, see, as I told you that these triazoles are displaying various kinds of biological activities like anti-cancer, anti-HIV, anti-inflammatory, anti-hepatitis, anti-diabetic, antibacterial, anti-tubercular, antifungal, Alzheimer, etc., as well as many more biological activities. Not only in studying therapeutic properties, but also their different kinds of polymeric properties, material science properties, people have studied. Okay, see, as I have told you that they are highly stable, used in different kinds of fields like biomedical imaging, as well as bioconjugation, as well as in drug discovery process for the design and development of different molecules to cure cancer or some infections. It may be bacterial, fungal, viral, whatever it may be. As well as I have told you that this click chemistry approach is used for the design and development of new materials, and those materials were studied for their different kinds of properties as well. And even though people have used different kinds of synthetic methodologies, okay, with this background, I will focus on how we have contributed in terms of click chemistry approach and how we have designed the molecules, simple molecules, and used the starting materials which are available in the postgraduate laboratory. And we have studied their therapeutic properties like antifungal, antibacterial, antioxidant, antitubercular, anti-inflammatory, as well as we have also studied their different types of theoretical properties through computational study as well. Okay, just a simple molecule we have considered over here. We have designed the molecule with the help of the TB enzyme, that is a DPRE1 enzyme, and see towards the structure how it is a simple small molecule having 1, 2, 3 triazole in their skeleton, which shows drug-like properties, as well as some ethynyl linkage in the structure. And this aromatic part which will control the lipophilicity of the molecule. Accordingly, we have designed, synthesized, and well executed, and later on we have characterized by means of their spectral properties and studied their therapeutic activities as well. And see the synthetic strategies, as I have mentioned that the starting materials should be commercially available, as well as it should be available in the post-graduation laboratory. See, we have undertaken the very first task from different types of phenols, highly substituted phenols. It may be chloro, methyl, nitro, ido, different types of phenols, subjected for alkylation reaction by using propargyl bromide in potassium carbonate in the solvent like DMF, resulted into the corresponding terminal alkyne. Simply alkylation reaction is here. Later on, another starting material is required to carry out this click reaction, that is azide. And these particular benzyl azides were prepared from corresponding bromides via nucleophilic substitution reaction with sodium azide. And those bromides are prepared from corresponding alcohols by using PBr3 or HBr. And those benzyl alcohols were prepared from corresponding commercially available benzaldehydes via reduction reaction by using sodium borohydride reduction. Means aldehyde to alcohol, alcohol to bromide, bromide to azide. In other way, the same azides can be made from this particular alcohol to mesylate or tosylate, followed by nucleophilic substitution reaction of sodium azide gives this particular benzyl azides. See, we have 2 different types of starting materials, 7 starting material related to alkyne, 5 starting material related to benzyl azides. And how these 2 types of substrates we have utilized to make different types of 1,4-disubstituted 1,2,3 triazoles. And see, now the simple reaction, that is a click reaction between this terminal alkyne and this benzyl azide by means of click chemistry approach using copper acetate as a source of copper to activate this terminal alkyne in the solvent like tertiary butanol water at a room temperature for 19 to 28 hours resulted into the 1,4-disubstituted 1,2,3 triazose. Means see how many molecules we have made. This is 7 alkynes, this 5 acids, 35 compounds, and all are new compounds. And see the structures of those new compounds having substitution on this ethyl linkage, means this phenol, as well as the substitution on these benzyl azides in terms of nitro group, chloro group, bromo, as well as the methyls. Different kinds of variations are there. After synthesizing all these compounds, 1,4-disubstituted 1,2,3 triazose, we have well characterized by means of spectroscopic techniques, that is H1NMR, C13, as well as the HRMS, and we have confirmed the structures of all these 1,4-disubstituted 1,2,3 triazose. Furthermore, we have screened these synthesized compounds for their anti-tubercular activity as well as antioxidant activity. We have performed this anti-tubercular activity against the strain like MTB S37RA and determined their MIC as well as IC50 values by using the standard drugs, that is rifamycin as an anti-tubercular drug. And see the red data indicates that some of the drugs are showing better anti-tubercular activity with lower MIC as well as IC50 values against the standard drug, that is rifamycin. Furthermore, see this particular table, third row indicates for IC50 values. Again, this third row, this row indicates for their IC50 values because the number of compounds are more compounds, as well as I mentioned that regarding the antioxidant activity for these synthesized compounds. And see the standard drugs, 2 drugs were used, that is butylated hydroxytoluene. They are the antioxidant agents, as well as ascorbic acid. And the IC50 value for these two, 16.5, 12.7. As compared to these 2 drugs, some of the compounds like 23E, then 22B, 22A, as well as 21A, here also some of the compounds are showing better activity as compared to the standard drug, that is a BHT as well as ascorbic acid. As these synthesized compounds are showing better anti-tubercular activity, we have studied theoretically with the help of molecular modeling study, molecular docking study, and we have docked these synthesized compounds, and this is the best example of most active compounds. We have docked again the active site of DPR1 enzyme, and this compound is interacting with the different kinds of amino acids and showing better interactions with lower docking score. See, in short, in summary, these are the most active compounds against the TB as well as antioxidant activity. Furthermore, we have screened these synthesized compounds against the cancer cell lines like THP1, A549 and PANC1 cell lines, and none of the compound is toxic, as well as we have analyzed these compounds for ADME properties also. And this particular work was published in the journal Medicinal Chemistry Communication for the year 2015. In this particular attempt, we have started the synthesis of the compounds like phenols, different kinds of phenols we have used. In our next attempt, just we have changed. Here we have used different kinds of benzyl acids. See, benzyl acids. In our next attempt we have changed the acid part only, and remaining part we have kept same. Means the alkyne part we have kept same, and we have varied this particular acid, and those acids were synthesized from corresponding anilines. We will see. Accordingly, we have designed the molecules and synthesized those molecules. As we have seen how to make alkynes, just I will tell you how to make such kind of acids from commercially available anilines. It may be plain aniline, toluidine, anisine, chloroanilines, etc. These kinds of anilines we have used. These anilines on chloroacylation followed by nucleophilic substitution of sodium acid resulted into the azidoamide formation takes place. Azidoamide formation, and these are the azidoamides. And later on, we have also carried out the optimization for this particular reactions. And in the optimization, we have used a copper acetate as a source of copper, as well as the medium we have changed in place of tertiary butyral water system. We have used some kind of ionic liquid, and see the drastic change. In earlier case, it requires 20 to 28 hours. However, here it requires only 7 hours through conventional method, means at room temperature. And we also performed the same experiment under ultrasound irradiation. Within 35 minutes, we have achieved the highest yield of the products, around 95%. And by keeping in mind this optimized reaction condition, we have used for making different kinds of 1,4-disubstituted 1,2,3-triazoles having this amide linkage in their structure. By using copper acetate, triethyl ammonium acetate as an ionic liquid, 1 ml we have used, and under ultrasound irradiation at room temperature for 35 to 40 minutes resulted into the such kind of 1,4-disubstituted 1,2,3-triazoles. And these are the structures of those triazoles analyzed by means of spectroscopy and screened for their antifungal activity against the 5 fungal strains, as well as anti-tubercular activity against MTBS37RB, as well as antioxidant activity. See, some of the synthesized compounds are showing equivalent antifungal activity. Equivalent, or sometimes some of the compounds are showing twofold antifungal activity. However, none of the synthesized compound is showing better anti-tubercular activity. However, some of the compounds are displaying better antifungal activity against the standard drug, that is the butylated hydroxytoluene. Okay. As these synthesized compounds are showing better antifungal activity, we have doctored these synthesized or most active compounds against the active sites of Candida albicans, lanosterol, 14-alpha demethylase, and showing better interactions with the different kinds of that particular amino acids or enzymes. And this work was published in the Journal of RSC Advances. In our next attempt, see, now we have varied the phenol part over here. In place of simple phenol, we have undertaken the task by introducing some acyl functionality in their structure. Because from astophenols we can make trillions of the compounds, like chalcones, then chalcones further converted into different types of heterocyclic compounds. And see, only we have introduced this acyl part, and the remaining part we have kept same. And we have designed such type of molecules and docked against the enzyme sterol 14-alpha demethylase, that is a fungal enzyme, and executed well by means of synthesis. Our synthesis starts from commercially available 3 phenols, that is plain phenol, paracresol, as well as parachlorophenol, subjected on acylation reaction, resulted into acetate derivative, followed by Fry's rearrangement. Our postgraduate students are regularly carrying out this Fry's rearrangement, and they are making such kind of orthohydroxyastophenols, and it gives exclusive only one product. One is a commercially available orthohydroxyastophenol, and just we have made 2 orthohydroxyastophenols from paracresol as well as from parachlorophenol, and it gives only one product because para position is blocked by means of those groups. And we have purified these compounds, synthesized compounds, we have purified by means of distillation and used further for making terminal alkyne by using propargyl bromide and potassium carbonate, same reaction condition. And furthermore, these alkynes, see 3 alkynes in our hand, and we have 5 terminal azides which we have made earlier under click chemistry conditions by using copper acetate tertiary butyral water solvent system for 20 to 30 hours, resulted into the 1,4-disubstituted 1,2,3-triazoles having this acyl functionality. And we are working on this particular ketone functionality for making thousands of the compounds, and we have synthesized around 18 compounds characterized very well by means of spectral techniques and confirm their structures, and followed by screen for their antifungal as well as antioxidant activity against the standard drugs like miconazole, fluconazole, as well as butylated hydroxytoluene. And some of the compounds are showing equivalent antifungal activity as well as antioxidant activity. And we have seen the binding interactions with the different kinds of 14-alpha demethylase cytochrome P51 as a fungal strain and showing better interactions with the different kinds of amino acids, and it is a fit in the cavity of that particular enzyme. Okay, in SAR, these are the most active compounds against the fungal strain as well as for their antioxidant activity, and this particular work was published in the Journal of Chinese Chemical Letters for the year 2016, and that time the impact factor of this particular journal was around 1.7, and now the impact factor of this journal is around 9. See, just simply in 6 years, how the impact factor of that particular journal has been changed drastically. In this particular attempt, we have undertaken the starting material, that is orthohydroxyastrophenone. Furthermore, just we have changed the source of phenolic OH group, because we can use that OH group to into corresponding terminal alkyne. See, we have designed such type of molecule, and C is equal to C, coumarin base. This coumarin is having a skeleton in most of the drugs available in the most of drug like warfarin, which acts as an anticoagulant drug. And this coumarin backbone showing different types of biological activities. And why not to incorporate this coumarin and triazole in a single molecular motif with the help of molecular hybridization approach, even though they are individually active. This triazole is individually active, this coumarin is individually active, but to see the properties by combining these two pharmacophoric groups in a single molecular motif by using molecular hybridization approach, design and screen such type of compounds against the two enzymes, that is DPRE1 enzyme as well as sterol 14 alpha demethylase enzyme, and design and synthesize very well from the commercially available resorcinol. This resorcinol on Pitchman condensation reaction with ethyl acetate under acidic condition, it gives corresponding 7-hydroxy-4-methyl coumarin. And this particular experiment we are giving to the postgraduate students of second year regularly, and every student is making around 1 gram of this 7-hydroxy-4-methyl coumarin compound. And after finishing their practical, just we are throwing in the dustbin. But the same compound we have undertaken and purified very well. We have done 2 to 3 times crystallization and used further for making such kind of terminal alkyne. We have only one alkyne in our hand, and we have carried out the click reaction with already synthesized benzyl azides in presence of copper acetate, tetrahydrobutanol, water, solid system for 17 to 21 hours at room temperature, resulted into the coumarin based 1,4-disubstituted 1,2,3 triazoles. Similarly, this 4-hydroxy coumarin on alkylation gives this terminal alkyne, and under similar reaction condition by using already synthesized benzyl azides gives corresponding 1,4-disubstituted 1,2,3 triazoles. Means we have 5 azides, we have utilized these 5 azides over here, and totally around 11 to 12 compounds we have made, and these are the structures of those coumarin based 1,2,3 triazoles, characterized very well by means of spectral analysis. And furthermore, we have studied their biological activity against for anti-TB, antioxidant, as well as antimicrobial evaluation like antifungal as well as antibacterial. We have screened these compounds for TB, and some of the compounds are showing a good anti-TB activity. Some of the compounds are showing good, better antioxidant activity, and almost all compounds except this 24D, almost all compounds are showing antibacterial activity as well as antifungal activity. And furthermore, we have talked the most active compounds, that is 21F, in the active sites of DPRE1 enzyme, because our synthesizer compounds are showing better antitubercular activity. These are the most active compounds having lower MIC value for TB, lower MIC value for antioxidant, IC50 value, and these two compounds are showing lower MIC value for antifungal as well as antibacterial activity. And this particular work was published in the journal Medicinal Chemistry Research for the year 2016. See, here we have used this coumarin. We have used this coumarin and made from commercially available compounds. In our next attempt, again, we have made different kinds of coumarins, different types of coumarin. Only the change is here ester part, and there is no methyl group. And that particular 27 number coumarin is made from again resorcinol. This resorcinol on Wilsmer-Huck formulation reaction by using POCl3 and DMF resulted into the hydroxyl substituted salicylaldehyde. This salicylaldehyde on novel condensation by using diethyl malonate under acidic condition, novel condensation followed by cyclization resulted into hydroxylated coumarin, and this hydroxylated coumarin on alkylation by using propargyl bromide in presence of potassium carbonate resulted into this terminal alkyne. We have one alkyne. We have already synthesized benzyl azides under clean chemistry condition by using copper acetate at room temperature resulted into the coumarin based 1, 2, 3 triazose. 8 compounds we have made, and these are the structures characterized very well and screened for their antifungal activity against the 5 strains as well as antioxidant activity. The standard drugs miconazole, fluconazole as well as butylated hydroxytoluene for antioxidant activity we have utilized over here. See, some of the compounds are showing equivalent activity or twofold activity against the standard drugs. We have talked these compounds against the enzyme that is 14-alpha demethylase and showing better interactions with the different kinds of amino acids, and these are the most active compounds. And this particular work is published in the Journal of Chinese Chemical Letters for the year 2016. Means how one can use the compounds made in the postgraduate laboratory. Okay. In our next attempt, just here we have used benzyl azides. In our this attempt, we have used benzyl azides here. But in place of benzyl azides, why not to use the azides derived from different kinds of anilines? And accordingly, we have designed the molecules. See over here, the coumarin part we have kept same. Just we have changed the azide part over here, and accordingly we have designed the molecules and synthesized them very well from commercially available resorcinol, as I already mentioned how to make this terminal alkyne, this terminal azide, and we know very well how to make such kind of azides from anilines on acylation, chloroacylation followed by reaction with sodium azide. And now we have a terminal alkyne, we have azide under clean chemistry condition resulted into the coumarin based coumarin as well as a triazole as well as amide, means 3 different types of pharmacophoric groups we have introduced in a single molecular structure. In second attempt, we have used the alkyne derived from 4-hydroxycoumarin on click reaction with azides resulted into the corresponding amide and coumarin based 1, 2, 3 triazose. Okay, these are the structures of those coumarin based triazose and evaluated for their antifungal, anti-TB as well as antioxidant activity. And the data for antifungal activities, some of the compounds are showing twofold activity as compared to the standard drug, and some of the compounds are showing equivalent activity. Furthermore, none of the compound is active against the TB strain, as well as none of the compound is showing better antioxidant activity except this 15A compound, which shows equivalent antioxidant activity. Okay, as these synthesized compounds are showing better antifungal activity, we have talked these particular compounds against the active sites of nanosterol 14-alpha demethylase strain and showing better interactions with different kinds of amino acids. And this particular work is published in the Journal of Arch Pharma, having impact factor around 4.8. In our next attempt, so far we have used different kinds of phenols. Now, why not to change one of the starting material, and that starting material or the core in that particular starting material should be available in different kinds of drugs. And here we have undertaken the task by using isatin. This is particularly isatin having NH group in their structure. Here triazole, which we have seen already and displays drug-like properties. As we know, this triazole shows better anti-tubercular as well as antifungal activity. We have designed this particular molecules against the enzyme of 14-alpha demethylase of Candida albicans and synthesized these particular compounds from commercially available isatin. This free NH group is there. This particular compound was subjected on base catalyzed reaction by using sodium hydride. N- formation takes place on alkylation with propargyl bromide gives this alkyl substituted isatin. This particular alkyl unsubjected with benzyl azides by using copper acetate click chemistry condition resulted into the isatin incorporated 1,4-disubstituted 1,2,3-triazose. Here benzyl azides were used. Why not to use aryl azides directly derived from corresponding anilines, that anilines on diazotization followed by substitution with sodium azides. This terminal alkyne and azide on click reaction in presence of copper acetate resulted into the isatin based 1,2,3-triazose. We have characterized them very well and screened for their anti-TB activity against MTBS37RA, antioxidant activity as well as antifungal activity. See here, none of the synthesized isatin based compound is showing better antioxidant activity. One or two compound is showing good antioxidant activity, and some of the compounds are showing equivalent antifungal activity. And we have talked about these compounds against the 14-alpha demethylase of Candida albicans because these synthesized compounds are showing good antifungal activity. And these are the most active antifungal compounds with a lower MIC value. This one is the best antioxidant activity with lower IC50 value. This one is the best anti-TB activity. These two are with lower MIC values are there. And all these synthesized compounds were screened for their toxicity against these three cell lines, and none of the compound is toxic, as well as we have analyzed for their ADME properties, and this particular work was published in the Journal of Heterocyclic Chemistry in the year of 2017. In our next attempt, we have changed one of the starting material. In place of isatin, we have taken here benzothiazinone, and remaining part we have kept constant because remaining part showing different types of biological activity as we know. We have designed such type of molecules against the enzyme DPRE1 and synthesized from commercially available orthoaminothiophenol. This orthoaminothiophenol on reaction with chloroacetic acid in presence of sodium acetate gives base thiazinone. This only one compound. This on alkylation with propargyl bromide gives terminal alkyne, and this terminal alkyne on subjected with benzyl azides resulted into the base thiazinone based of 1,4-disubstituted 1,2,3-triazose. In place of benzyl azides, here we have used aryl azides on reaction with this base thiazinone based alkynes under same reaction condition resulted into the base thiazinone based 1,2,3-triazose. We have these are the structures of all the synthesized compounds characterized very well by means of spectral techniques and analyzed for their anti-tubercular activity against the two strains of TB, that is MTBS37RA, M. bovis BCG strain, as well as antioxidant activity. And see here, some of the compounds are showing good anti-tubercular activity as well as good antioxidant activity. As these synthesized compounds, we are talking against the enzyme of DPRE1 and showing better interactions with different kinds of amino acids, and these are the most active compounds with lower MIC as well as IC50 values, and all the synthesized compounds were screened for their toxicity against these three cell lines, and none of the compound is toxic. This particular work was published in the Journal of Bioorganic and Medicinal Chemistry Letters for the year 2016. In our next attempt, we have changed one of the starting material. In place of base thiazinone, why not to introduce thiazolidinone, because thiazolidinone, this particular core is available in various anti-diabetic drugs and showing anti-diabetic activity as well as anti-inflammatory activity. As we know, the triazole showing different types of antimicrobial, antifungal, antioxidant, anti-TB activity. Accordingly, we have designed and synthesized this particular compound from this is thiazolidinone and aromatic aldehydes. We have used only 3 aromatic aldehydes. On novel condensation with this thiazolidinone resulted into the arylidine thiazolidinone, and this arylidine thiazolidinone on alkylation with propargyl bromide gives propargylated thiazolidinone, and this propargylated thiazolidinone were subjected for click chemistry by using already synthesized benzyl azides resulted into the thiazolidinone based 1,2,3-triazose, And these are the structures of all these thiazolidine based 1, 2, 3 triazoles characterized by means of NMR as well as a mass, and screened for their anti-inflammatory activity, antioxidant, anti-TB activity. See here, all the compounds are showing a good anti-inflammatory. However, the two compounds are showing better or excellent anti-inflammatory activity as compared to the standard drug diclofenac sodium, showing EC50 value is a 0.72, our synthesized compound 0.84, 0.64, 0.70, means equivalent antifungal, anti-inflammatory activity. Some of the synthesized compounds are showing better antioxidant activity. However, none of the compound is showing anti-TB activity against the two strains of TB. Moreover, we have also studied antimicrobial activity against the bacterial strain as well as the fungal strain, and almost all the synthesized compounds are showing better antibacterial as well as antifungal activity. As these synthesized compounds are showing anti-inflammatory activity, we have doctored the most active compounds against the active sites of PPR enzyme and showing better interactions with that particular enzyme. And just I want to show you the most active compounds. So this one is most anti-inflammatory activity showed by these two compounds, and these two compounds are showing good antioxidant as well as antimicrobial activity. This particular work is published in the Journal of Polycyclic Aromatic Compounds, and we have also published one Indian patent on this particular chemistry. In our next attempt, why not to use a drug, as we have seen that INH is showing, or INH isonicotinic acid hydrazide is prescribed by means of doctor to show better to cure the TB. And accordingly, we have designed, why not to introduce that INH nicotinic acid hydrazide as well as para-chloro-para-methoxy para-nitrobenzoic acid hydrazide, and to make hydrazoles of different kinds of aldehydes having 1, 2, 3 triazole skeleton in their structure. See, the chemistry is very simple, commercially available 4-hydroxybenzaldehyde unsubjected with propargyl bromide resulted into the alkylated aldehyde, which are further subjected with click chemistry by using benzyl azides resulted into the triazole based aldehyde, and this aldehyde on reaction with acid hydrazoles based on nicotinic acid, isonicotic acid as well as para-nitrobenzoic acid hydrazide at a room temperature gives corresponding hydrazoles, and a total 12 compounds we have made. And see, these are the structures and screen for their anti-TB as well as antioxidant activity. And here we have perfectly studied the structure activity relationship. This red compound showed better anti-tubercular activity as well as antioxidant activity. This red compound 20A, 20D, 20G and 20J, they are derived from INH, this drug, and some of the compounds, see, the drug is showing MIC value or IC50 value, and our synthesized compounds are showing equivalent anti-TB activity with lower IC50 as well as IC90 values. Means only compounds derived from INH are showing better anti-TB activity as well as antioxidant activity. We have further studied for their docking interactions with the enzyme, that is DPR1, and showing better interactions, and these are the most active compounds. Furthermore, we have studied their toxicity against the 3 cell lines, cancer cell lines, and none of this synthesized compound is toxic. Furthermore, we have analyzed for their ADB properties, and this particular work is published in the Journal of Mineralogy and Medicinal Chemistry, having impact factor around 3.3. Okay, in our next attempt, in place of para-hydroxybenzaldehyde, why not to have ortho-hydroxybenzaldehyde or salicylaldehydes? These salicylaldehydes were subjected on alkylation with propargyl bromide and reaction with benzyl azide by using click chemistry condition resulted into the triazole based aldehydes, and these triazole based aldehydes were subjected on hydrazone formation by using INH based hydrazide, isonicotinic acid hydrazide, nicotinic acid, para-nitro, para-chloro, para-methoxy, benzoic acid hydrazide, and these are the structures of all these hydrazides. There are about 35 compounds we have made, and see over here we have screened these compounds for their anti-TB activity against the 2 strains antioxidant activity also. See, the red data showing better antitubercular activity against the two strains of TB, 24A, 24F, 24K, 24P, as well as, see here, 25A, 25E, 25H, 25M. All these compounds are derived from the corresponding aldehyde as well as isonicotinic acid hydrazide and showing equivalent or somewhat better antitubercular activity with lower IC50 value, that is a 0.09. And these are the structures of those highly active compounds. And see, in their structure, INH is there in all their structures. INH as well as the three compounds are having bromo substituent in their structures, and none of the synthesizer compound is toxic against these four cell lines. And furthermore, we have studied the properties, ADME properties, and we have communicated this particular work to the journal, and very soon we will receive the acceptance letters, final acceptance letter for this particular work. In our next attempt, see here, why not to introduce some different kinds of pharmacophoric group like a quinone based as well as this ethyl linkage triazole compounds. And we have designed this particular compounds and started synthesis from commercially available meta-anicillin. This meta-anicillin on acylation reaction gives acetylide. This on Wilsmer-Huck formulation reaction gives quinone based aldehyde, that is 2-chloro-3-formylquinone. This 2-chloro-3-formylquinone on nucleophilic substitution reaction with sodium azide in DMF gives tetrazoloquinone aldehyde. This aldehyde on reaction with sodium borohydride gives alcohol. Alcohol further reaction with methyl chloride followed by nucleophilic substitution reaction with sodium azide gives corresponding tetrazoloquinone azide. We have made only one azide over here, and already synthesized alkynes derived from phenols. One azide, this 8 alkynes which we already synthesized on click reaction gives corresponding 9 compounds, total 9 compounds of tetrazoloquinone based 1,2,3 triazose. Not only that alkynes derived from phenols, but also simple phenylacetylene is also used to study the structure activity relationship, gives corresponding 1,2,3 triazose. These are the structures starting from particular azide and triazose, and the azide as well as all the triazoses were screened for their antibacterial as well as antifungal activity. And see here, the azide does not show any kind of antifungal as well as antibacterial activity. However, our synthesizer compounds are showing equivalent or twofold or sometimes threefold. Here, see, this 23H is showing 4 MIC value, and the standard drug kanamycin or amphotericin B, they are showing 16 microgram per ml. See, means our compound is 4-fold active, and some of the compounds are 2-fold, some of the compounds are equivalent activity as compared to the standard drug like different types of antifungal as well as antibacterial agents. As they are showing better antifungal activity docked against the active sites of sterol 14 alpha demethylase and showing better interactions with our synthesizer compounds. And these are some of the most active compounds with lower MIC values. And this particular work was published in the journal Polycyclic Aromatic Compounds. So far, we have studied monomeric compounds. Monomeric means we have introduced only one triazole ring in their structure. And now we will see dimeric compounds, dimers having 1, 2, 3 triazoses, and how these dimeric compounds can be synthesized by using click chemistry approach. And as our body, human body, is a totally cytosymmetry, and on the basis of our human body, we have designed the molecules having cytosymmetry in their structures. Like here, see. This kind of monomers means this part is alkyne, this part is azide, and made such kind of 1,4 disubstituted 1, 2, 3 triazoses. Here some linker is there. This part, these two parts we can consider like this way. We can make different types of dimeric triazoses. We have designed the dimeric triazoses based around phenols diphenols we can call it, and monomeric form of azides we can use. And see, we have studied there are different types of properties, like anti-tubercular, anti-cancer, antioxidant properties, and accordingly we have designed the molecules and synthesized from commercially available starting materials like aromatic amines. As I already mentioned about how to make this amide-based azides, these anilines were subjected on fluoroacylation, followed by reaction with sodium azide gives azidoamides. And see this resorcinol or catechol, resorcinol and catechol on dipropargylation by using two equivalents of propargyl bromide under same reaction conditions. This particular terminal alkynes, bis alkynes were made, and these synthesized bis alkynes derived from resorcinol or catechol on click chemistry condition by using azidoamides gives corresponding 1,4-disubstituted 1,2,3 triazose, and not only this one but also in the dimeric form. In the dimeric form means 2 triazole units are there in their structures. These are the structures of all the molecules, characterized very well by means of spectral analysis and screen for their different kinds of biological activity, like anti-tubercular activity against the standard drug like ethambutol and ciprofloxacin. The only two compounds are showing a good anti-tubercular activity as well as antifungal activity against the 4 strains, and the bold data indicates that most of the compounds are showing better antifungal activity, as well as most of these synthesized dimeric triazoles are showing better antioxidant activity. Furthermore, we have also performed anti-cancer activity against the 3 cell lines, that is MCF7, A549 and MCT10A, and some of the compounds are showing better anti-cancer activity as compared to the standard drug, that is cepatinib, 2.97 MIC value, but our synthesizer compound is 2.80. Here also, against this particular strain, the 2 compounds are showing better anti-cancer activity. Okay, see, this is the somewhat a structure activity relationship and most active compounds I have shown over here. This particular compound is showing better anti-cancer activity. This particular compound is showing better anti-TB. This one better antifungal, and this compound is showing better antioxidant activity. As I have told you that some of the compounds are showing better anti-cancer activity, and we are doctored better antifungal activity. We are doctored these synthesized compounds against the sterol 14 alpha demethylase, which is a fungal enzyme, and showing better interactions. As well as we have doctored these particular compounds against the active sites of EGFR enzyme and showing better anti-cancer activity. And this particular work was published in the journals, that is the New Journal of Chemistry for the year 2021 as well as the BMCL 2019. In our next attempt, just we have changed the amide. We have changed the azide part, and in place of aromatic skeleton in their structure, we have used aliphatic skeleton, and those aliphatic azides were made from corresponding amines via fluoroacylation followed by substitution. And these same alkynes, these bis alkynes, we have used by using click chemistry condition using copper sulfate and sodium ascorbate in pH 400 resulted into the dimeric triazoles. These are the structures of those dimeric triazoles, and screen for their anti-TB activity, and some of the compounds are showing better anti-TB. Antifungal activity, most of the compounds are showing twofold or equivalent antifungal activity against the fungal strains, as well as some of the compounds are showing better antioxidant activity with lower IC50 value. And these are the most active structures and doctored against the sterol 14 alpha demethylase and showing better antifungal, better interactions. And this particular work is published in the Journal of Heterocyclic Chemistry. Now, in our further attempt to make a dimeric triazoles, only we have changed the linker over here or spacer. In place of phenols or resorcinol or catechol, we have undertaken the task by using piperazine based dimeric triazoles. And accordingly, why piperazine? Because piperazine based drugs are available in the market like ritamicin, and we know about the triazole. See, As this piperazine, two NH groups are there, and which on this alkylation by using propargyl bromide gives a terminal alkyls and already synthesized amide-based acids on click reaction by using copper sulfate, sodium ascorbate resulted into the piperazine-based. This is the some spacer or linker piperazine-based 1,4-disubstituted 1,2,3-triazose, and these are the structures of all these piperazine-based dimeric triazose, characterized very well by means of spectral analysis and screen for their biological activity like TB, fungal, as well as antioxidant activity. And some of the compounds are showing better anti-tubercular activity as well as antifungal activity as well as antioxidant activity. And this is the structure most active compounds and showing better interactions with the fungal enzymes, and this particular work has been published in the Journal of Synthetic Communications. With this, I'm very much thankful to the management of Bharati Vidyapits as well as principal and faculty. And the most important thing, I would like to appreciate the work carried out by the three students, Dr. Mubarak Sheikh, Dr. Tejasri Deshmukh, and Dr. Satish Akulkar. Some part of their works I have presented over here. Without taking the support from different collaborators from NCL Pune, then Vishwakarma University Pune, Central University of Gujarat, as well as... yes, sir.
No, sir, sorry. You will continue.
Yeah, as well as without financial supports from different funding agencies like CSR, UGC, DST, as well as Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. And I would like to dedicate this particular lecture to the Nobel laureates for the year 2022 for their great contributions in terms of click chemistry, that is Professor Bartolozzi, Professor Meldon, Professor Sharpless, and all of you people have listened patiently. Thank you, one and all. Thank you. Over to you, sir.
Thank you, sir, for giving valuable information about click chemistry to all of us. Now I invite to Dr. T.R. Lohar, sir, for giving the vote of thanks. Dr. T.R. Lohar, sir.
Prior to that
Thank
one,
you,
if
madam.
you are having some queries, questions, you can ask me.
Sir, we are conducting this lecture in a broadcast manner. So we are able to conduct the question and answer session.
Okay.
Thank you very much, madam. First upon, I am very much grateful to our respected principal, Dr. D.G. Concessor, for their constant support, guidance to arrange such type of activities in our college. I am also grateful to today's chief guest, Professor Shingate sir, for giving nice lecture on importance of triazole chemistry in medicinal field. In his talk, sir mentioned the importance of click reactions. That is, in click chemistry, how molecules combine together to form triazoles. Sir has published number of research papers in well-reputed journals. I am also thankful to faculty members and the students of our college for attending this webinar. Now, by taking the permission of respected principal sir, I declare that this webinar is over. Thank you very much, sir.